Hi folks, we're here to talk about Chapter 7, Microbial Growth, and I want to walk you through this PowerPoint here for this recorded lecture. I told you guys that I would actually do this, and so hopefully you'll find this interesting, and hopefully this will help you out as you're going through for this weekend as you guys are preparing for your exam. Okay. So again, mention microbial growth. And we're going to look at really the basics first. We'll really take a look at the basics of microbial growth, and then after that point we'll go into a couple other fundamental properties that I really want you guys to kind of get a handle of when looking at growth of microorganisms. And really what that means is to understand how growth works is really important, because when you know how a microbe works, what conditions are set in place for that microbial organism to grow, to actually divide, undergo replication, then you know a little bit more of how to treat it. And that's really what that means to you as clinicians later on in your career, is that you are actually going to look at and understand the infections and how they work, because you understand, you have a good background hopefully from this chapter on the fundamental basics of growth in an organism. Okay, so we'll start off here. You know, when we have nutritional requirements being met, and I'll go ahead and use this pen to kind of highlight some things that I think are important for you to kind of grasp here. Not that you don't need to read through them all, but if you're looking at some highlighted points, some hot bolded print, these are important things to understand with, right? Looking at the first bullet point, when nutritional requirements get met, a microbe will again enlarge in size, right? So we see an enlargement in size, right? Sorry about this. This is trying to work out this pen here. Enlargement in size, and will eventually divide. And dividing basically means what? Binary fission, right? Binary fission. That's really what we're talking about here. Undergoing replication, undergoing asexual reproduction, in fact. So when all conditions are met, all nutritional conditions are met, and let's say in an experiment you give the organism all the nutrients it needs, the organism will then enlarge in size and eventually undergo replication. And that's really the whole goal of a microorganism, is to gain enough nutrients to actually undergo replication. And that's really all it cares about. Does not have any other purpose other than that. Otherwise, you know, other than actually entering cells, harvesting nutrients, and then taking those nutrients for replication. That's really the whole goal of what we're talking about here, right? So what is microbial growth? Microbial growth is cell division, right? Okay. Produces new daughter cells. These are new replicated cells through the process of mostly binary fission if you're talking about a bacteria, right? Increases, the key word is increases total cell production, right? This is really meaning gaining more daughter cells, gaining more clones of themselves, and actually increasing in numbers, right? That's what microbial growth basically means. In a larger scale, when you look at microbial growth in a tube, test tube, and a petri dish, and any other format we're going to continue to look at during lab, for example, we're going to see growth and growth and growth on a large scale. If it's big enough for us to see for example, on a test tube or on a plate, then that means there is a tremendous amount of growth of that organism or organisms in for example, in a media, okay, in a solution. So most of them are studying about bacteria, about microbes in general, not only bacteria but fungi and the like, really come from actually culturing these in the lab, growing them in a lab and studying how they actually grow themselves. And we know that, you know, less than 1% of all the species out there in our planet is actually, you know, come from actually studying them in the lab, right? And we still don't know the other 99% because we haven't had a chance really to, or know how to actually grow these in the lab, right? Usually in a lab, the bacteria get grown as a pure culture. So one bacteria gets inoculated one single species gets inoculated, and then we see the growth over time, right? But in nature, the reality is that bacteria intermingle with other bacteria and other types of living organisms like archaea and eukaryotic organisms. So it's a big community in the environment, whether that environment being soil, water, other living organisms. These are environmental areas where actually all of them will live. Okay. So the environmental factors, and this is the key word to understand. This is the key statement. Statement number 3 is a key statement to understand going forward, because you got to keep this in mind. The environmental factors play a big role, a huge role in metabolism and the structure of bacteria. As I've mentioned, bacteria grow in environmental areas. They've been here for a very long time, longer than humans have been on Earth. And because of that, they've actually adjusted to the environment. And because of that, they actually adjusted their cell structure, their metabolism to meet those needs in the environmental area in which they live. And this is where we're going to have diversity. We're going to have different types of microorganisms found on Earth, with as far as metabolistic properties and structures. You got to keep that in mind when we're talking about these different types of species there. Okay, cells that live in a biofilm. We've talked about biofilms before, but cells that live in a biofilm communicate and collaborate to survive. It's one of these really interesting places that you see in microbial growth, in which there is, normally these species are kind of brought to their own cells in the environment, but in this area they actually live as a community. They actually count on each other for survival. So this is really interesting. Now, what do I mention? Biofilms, of course, I've mentioned this before, but biofilms are notoriously difficult to treat. Really, really difficult to treat because biofilms create a matrix. This solution that, well, solution, but this chemical composition that the bacteria start to secrete acts very much like a huge slime layer. It helps them to attach to themselves and to a surface, and that's difficult to pull off, completely take off of surfaces like hospital beds, hospital door handles, rails, hospital equipment, supplies, things like that. So it's a big concern. In those biofilms, you might have something that's pathogenic, something that actually causes harm to a human, and that's why it's actually a big concern, because those can cause infections, and those infections can be persistent infections. Okay, so here, this is figure 7.1, and 7.1, you actually look at it, you're seeing a good example of a biofilm on a catheter. It's actually inside the catheter. It's on the outside of the catheter, but inside the catheter. And this is actually because we've got, for example, many types of microorganisms coming from the bladder surface and from urine itself. Urine is not a sterile substance, and actually have collected, and it started to adhere to the inside of the catheter. So actually what you see here is the inside portion of the catheter. They've cut the catheter and looked inside and actually seen the biofilm. This is under a electron microscope, right? So this alkagene species, these little dots here, in fact, and some of them are coccus, but you can also see them are paleomorphic. They actually can actually cause blood infections, and so this is a big issue, for example, when you're talking about this, right? So bacteria usually divide by binary fission. That's usually most organisms will divide, that are bacteria, divide by binary fission, but there is other ways to undergo replication. And again, most prokaryotes undergo binary fission, but in the event, there are some other species that do other types of replication. Okay, so we've seen this before, right? We've seen this, the steps of binary fission, same ones as before. We've seen these where you see the genetic material replicating, the cell growing in size. We start to divide that cell into two, and then you create a separate clone, an exact copy of bacteria. And in fact, this is E. coli. So this is one E. coli, and actually not one, but two E. coli microorganisms, bacteria. And that arrow represents the dividing, I'll draw that there, the septum, if you will, dividing and creating two separate E. coli bacteria, right? So you see that there. Okay, so here's the other process of binary fission. In fact, this is called budding, okay? And budding is a type of asexual reproduction, not sexual, but asexual. And it kind of falls in the same process of binary fission in the fact that the cell will elongate, will grow in size, but instead of actually forming two separate plasma membranes and cell walls, it actually has a smaller portion of the cell wall and plasma membrane that start to kind of bud out, if you will, bud out. And so any of the chromosomes that are made in the original, for example, the original bacteria, or we use this sometime in fungi because fungi do this, they duplicate the chromosomes and they stick that into the actual bud formation. The separation occurs, and the smaller bacteria or fungi, for example, will cleave off and grow in size, and they'll actually grow to a normal size, and then these species will become other species, other individuals in the same process of budding. Okay, there are some bacteria and fungi that actually spore, if you will, and this is very similar to actually budding in the fact that there is a smaller version of the particular species that breaks off, becoming a spore. Spores are really interesting because spores can actually fly through the air. They can float in air. They're so light that they can actually be found in air and in water as well, which can be difficult to treat because if that spore in fact lands somewhere else, that spore can actually grow into more fungi and more bacteria. So we talk about fungi because fungi do this a lot, forming spores, but some bacterial species do this as well. Streptomyces is a type of fungi genus that actually does this as well. And endospores are kind of a spore, they fall in the spore formation group. Okay, so a big term here to discuss, right? Generation time. Generation time is the time it takes for one cell to divide into two. And we talk about this being the time it takes for one bacteria to turn into two. That is a really, really diverse time frame. Not every bacteria has a certain time frame, and that's not all standard. So some bacteria, for example, as it says there, 15 minutes will you'll see one bud into two, or excuse me, will, because of binary fission, become two, up to 24 hours and everything in between. Depends on the species and depends on conditions. And what do I mean by conditions? Environmental conditions. Conditions that are set up to provide the nutrients. Again, you have nutrients or you have the lack of nutrients, that's going to change growth or generation time as well. So that's a factor to play that play a role in generation time. Okay, so here's generation time, and a great example of that. You're not going to need to calculate generation time per se in the test, but you need to understand how this actually works, right? And so what we're looking at here in the figure 7.4, is one E. coli bacteria at the beginning, and through 3 generations actually become 2, 4, 6, 8 bacteria in the span of 1 hour, and you can see that 60 minutes, right? And so what they're asking you here is what is the generation time for E. coli? If looking at 1 goes into 8 in 60 minutes, and there's 3 generations, what's the generation time? So they figured that out for you. So if you take, you know, 60 minutes, how many minutes in 1 hour, and divide that by 3, because they generated 3 times in 60 minutes, the answer becomes 20 minutes. That E. coli, in fact, will have a generation time of 20 minutes. Okay, so looking at generation time, you see 1 here, 1 becomes 2, 2 become 4, those 4 become 8, and as I mentioned, if you let that go another 20 minutes, it would become 16, then 32, then 64, and etc., 128, and it keeps going and going. As long as you leave it alone, don't change environmental conditions, it will replicate indefinitely. Will continue to replicate through infinity, theoretically, so it'll keep going. And that number will grow as every 20 minutes in this example. Okay, so again, the availability of nutrients is a big factor in generation time. Have the number of nutrients there that the organism needs to survive and to grow, in fact, and replicate, then that will determine how fast it does that, how many times it does that, right? And so again, just to kind of show you, it varies. We see E. coli being 20 minutes, right, for example here. If you look at this one, tuberculosum, Mycobacterium tuberculosis, okay, and that is TB, right? Call that TB, right? Tuberculosis. 15 to 20 hours, really long time. And if you know about this disease, you know it is a very slow grower. Somebody that has TB, it will take a long time to generate symptoms of tuberculosis, and for the patient to gradually become ill and ill. So it's a very long-lasting disease there. Okay, so this is the growth curve for growth, right? So we look at a bacteria and we're looking at the growth there. Okay, so let me walk you through this figure here, figure 7.5. We see logarithmic growth in one part of the graph, right? And we also see logarithmic decline as well. And so we see this in this experiment. So in our first beaker here, our first flask, if you will, we've got a media that has all the nutrients that this organism might need. We've introduced organisms into the media and watched to see what happened. Okay, so we watch to see how growth is occurring. And so those blue dots represent the cells of the bacteria there in question, whatever that bacteria might be. And then looking at the second flask, we see, of course, that the number of bacteria represented by the blue dots have increased, right? And then we see in the third part of the graph where the beaker has a bunch more blue dots and indicated by the number of bacteria there. And then the fourth one, we notice something very interesting, right? We notice that there are less now bacteria than you see in the third flask, and you start to see this kind of blue line underneath. And as it mentions, it's all the waste and dead cells or dead bacteria there. Okay, so something interesting is occurring here. So let's look at the graph here. In what we call the lag phase, I'll stop right there, lag phase. In the lag phase, the cells are adjusting to their environment. So you don't see any growth occurring here in the lag phase. How do I know there's no growth occurring? Because the line has stayed stationary, straight. If there was growth in this portion of the growth curve, you would see the line move up. Since you don't see the line move up, that means there's no growth occurring. So the cells are adjusting to the environment. They're more than likely gaining nutrients, taking in nutrients from their environment in the process of preparing themselves to undergo binary fission and to create new bacteria. And in fact, the second part of the growth curve shows log phase, goes all the way up, right? And look how it shoots straight up. We call this logarithmic growth. The 1 turns into 2, 2 to 4, 4 to 6, 6 to 8, and obviously there's not just one bacteria, there's several there. So each one of those is undergoing that same generation time, and you see logarithmic growth, doubling every single couple minutes or however long this might be generation time for this particular species. And you see this growth and growth and growth. You see the graph go all the way up to the very top, okay? And we call this the log phase. And then you see the graph level off, right? The graph levels off because the number of cells dividing equals the number of cells dying. So they're limiting out in the amount of nutrients available. They've maximized the amount of bacteria that can be handled in that one particular flask. So remember that they're using up nutrients. And so since this is a closed flask, there's no extra nutrients being fed, this is going to limit out how much nutrients are in the solution. So as they grow, they consume more nutrients and more nutrients, and they kind of max out the amount of nutrients. So some of them will eventually die. And as they are dying, they're equaling in the number of growth, right? So for every 5 that grow, 5 are dying. For every 10 that are growing, 10 are dying. 15, 15, whatever number that might be, it's the same rate of growth versus death. Okay? And the final phase, you see what we call the death phase. And in the death phase, all the cells are dying. Okay? So we're seeing more death than we are dividing. There's more death than dividing. So we're, again, that's shifting the amount of dead versus living. So because we're seeing that, we're seeing more death occurring. And in fact, the built-up of waste and dead cells are because those have actually settled to the bottom. There's many more than what you just see interspersed inside the fluid. And so there'll still be living ones still surviving, still taking up the last bit of nutrients, but most of them have died, and we call this the death phase. Now, why is this really important to understand is because we also look at this in a living organism, like humans. When we see an infection occurring, and let's say a bacteria is infecting us, we see this coupled. You can actually take this and you know, match it right up to the inclination, the rise of signs and symptoms of an infection, and then the fall of those signs and symptoms. Or you can also lay this one right on top of being sick or feeling sick and then feeling better. So this goes hand in hand with the growth that's occurring, because these bacteria are growing, and when they're replicating, we see severity, right? We see the signs and symptoms in us getting sicker or the patient getting sicker. But then in the other part of that graph, it will eventually, go down for the most part. Some will stay constant, and then will cause the person to expire in an infection like that. Okay, so these two slides show you each of the phases, so I'm not going to go into detail with these. This is kind of, I talked you through this, but it's here for you if you want to look at this. Okay. Here's what we call a chemostat, and in fact, a chemostat is what we use in an industrial aspect, in an industrial lab, in industrial types of food processes, and what you see here is fresh media coming in, okay, fresh air coming in, old air going out, and old waste and cells going out. So constantly in this solution, you're having nice, always fresh solution and air, so that way it enables then the bacteria to keep growing and growing and growing. If you're left it like this, if you continue to do this, the bacteria will just grow and grow and grow infinitely, so they don't stop. If fed enough, they will actually do this, right? And what we're seeing here more than likely is yeast, right? So you can actually grow yeast this way. We use yeast for many things in industrial food and medicines and things like that. We use yeast a whole bunch. So this picture right here with this gentleman is a chemostat. They're making a lot of yeast. Okay. All right. So we're going to look at prokaryotes and their growth requirements here. We'll look at these basic growth requirements, and again, these mirror right into environmental stuff as well, right? Okay, so all microbes, right, on Earth find a niche, right? A niche is a particular place that or particular conditions that an organism prefers to live in, right? They adapt to those particular conditions. And these are all earthly or environmental conditions, right? Temperature, pH, types of nutrients, and even another one that we call O2, right? Oxygen. Oxygen is also a specific condition. All of which, depending on where you find the organisms on Earth, are where you find these different environmental conditions. Or you can flip that around. These in different environmental conditions is where you find different microorganisms because they have adapted to those areas. A lot of species have died along the way in the history of life, but the ones that have stuck around are because they've adapted to those areas. Okay, so talking about temperature, right? Really important for microbial growth and actually all living organisms, right? Temperature is really important. Okay, so usually this is the case, right? If you have a low temperature, that usually decreases enzymatic reactions. So enzymes don't work as well if you have a low temperature, and that's a general statement. Low temperature equals less enzyme function. At an increased temperature will usually mean increased enzyme function, right? Increase the growth rate, usually speaking. So this is a very general statement. Doesn't fit for every single organism. And usually when that temperature is too high, and again, there's not a set point because it depends on the organism. High temperatures usually denature proteins, right? Those proteins can actually be enzymes, but they can also be structural proteins, and we remember that proteins do everything for the cell. So when you start to denature these, they in fact will not work, will not activate. Okay, so when we talk about a protein being activated, it will usually have a shape to it. So let's say that the shape is like this. This line represents all of the amino acids bonded together, and we hold the shape just like this because of these red lines. These red lines represent bonds in between the amino acids, making this nice kind of helix shape, right? Everybody see the helix there? Okay, this shape there. So what occurs, and these red lines again are the bonds that hold the amino acid up and make it a nice shape. The shape means it's active, it can be used, right? What will happen in high temperatures is high temperatures will cause these bonds to break. Okay, these bonds to break, these bonds to break, and all these, this bond, this one, and this one. And what we talk about denaturing is actually that, right? Let's say that line right there falls apart, and because the bonds are broken, it's just one long strand of amino acids, not actually shapen like it is there because the bonds fall apart, because high temperature can break those bonds. And that's really what occurs, is the amino acid chain just kind of just creates a chain, does not hold the shape whatsoever. Okay, so here's three major temperatures that you see in the growth of a living organism. What we call the maximum temperature, okay, the minimum temperature, and the optimal temperature. Maximum, minimum, and optimal. The maximum temperature is the highest temperature that supports growth of an organism. So we got to be specific. Which organism are you talking about? So that's going to depend, right? So depending on the species, they will have a different maximum temperature. The minimum temperature is the lowest temperature that can still support the growth of that organism. Again, it just depends on what species you're talking about. That's the minimum temperature. And the optimum temperature is where cell growth is the highest. So it doesn't necessarily, optimum doesn't mean maximum, or optimum doesn't mean minimum. It just means optimum. A set point, either between, it's probably more than likely, you know, when you really look at this, it's going to be between the max and the minimum, right? Between the max and the minimum. If this is the max okay, temperature, and this is the minimum. Kind of impressed I can actually write letters on this thing, so I'm impressed there, right? So um, let's say the optimal temperature would be, you know, right here, optimal. Somewhere between the maximum and the minimum, whatever those temperature ranges are going to be, that's optimal temperature. The good thing is the scientists have figured out pretty much the optimal temperature of a lot of organisms that we're already studying. So they know the optimal temperature of growth. And for example, most bacteria usually grow about 37, 35, 37 degrees. And if you've noticed, we have that big incubator in the lab. That incubator is actually set at 35 degrees Celsius. That is the reason why, because that's optimal temperature for most organisms. Now, if we were scientists and we found, oh, a new organism that we weren't sure of, and we would want to test what temperature is the best growing for that organism, well, we would grow that organism or test organism growth in different temperatures. So let's say, you know, we put it on a petri dish or a media plate and put it in the 35-degree incubator, and then we would leave one out in the lab in that temperature, which is about, you know, let's say about 30 or so, maybe 25. And then we take it to the 30-degree incubator, and then we, you know, put it in a really cold temperature and watch to see the growth. And then we would take all four of those, let's say, and match them up, and we would say, okay, you see more growth on the plate here. That probably means because the optimal temperature is the 30-degree incubator versus the other ones. That's kind of how we can, you know, research is done with that. Okay, so here are the growth ranges of these prokaryotes, talking about these, you know, maximum minimums and these places where our optimal temperatures are for different types of prokaryotes, right? And so we use these terms here, and you can see these terms here, thermophiles, extreme thermophiles, excuse me, thermophiles, mesophiles or mesophiles, psychrotrophs, and psychrophiles. And you can see the temperature ranges there as well, right? All of those there are the temperature ranges for each of these ranges on the thermometer, and we can actually classify organisms based on those ranges. So we have organisms that are extreme thermophiles, mesophiles, psychrophiles, and psychrotrophs and psychrophiles. And so here are the temperature ranges. Make sure you know the temperature ranges and what classifies as a psychrophile, psychrotroph, mesophile, thermophile, and extreme thermophile. So they're very interesting to note there. The one that grows the best here for most organisms is mesophiles. Most organisms are in the mesophile range. And why the mesophile range? Because most temperature around the world is about that range for most parts of the year, and that's where you see more growth. Okay, usually in physics, you usually see this condition here, right? High temperature is directly, or temperatures are related to pressure, right? Temperature and pressure are directly related. Okay. Usually when you increase the temperature, you increase pressure. When you decrease temperature, you decrease pressure. They're directly related. When you put one up, the other one goes up. Put one down, the other one goes down. So usually this is, you know, coupled together. And so we talk about barophiles being able to withstand high pressures. And so we see high pressures forming in certain parts of the world. For example, in the deep sea, you have an extreme high pressure, and you even have microorganisms living in those areas. Mostly they are archaea, but you do see some new species of bacteria that are found in these high pressure deep sea environments called barophiles. There. Okay, so neutrophiles grow best in the neutral range, and that pH is between 5 and 8, right on the pH scale, almost by 7. 7 is the neutral, right in the middle. between 1 and 14, and 5 to 7 is kind of around that range. And we call these neutrophiles. And most of your organisms fall in the neutrophile range, right? Neutrophile range. Actinophiles are the opposite of the acid. They're the basic, right? So here's the range there. This is in the scale, the pH scale, 9 to 11 is the basic, right? So that's actinophiles. And what we're seeing here on the right side is what we call a soda lake, right? Okay. This lake is in California, in fact. This is figure 7.8. And so this is in California, in fact. And these long kind of pillars here, by the way, are limestone. Okay. Very acidy. This is caustic, right? We call this caustic, but this is very basic, right? Limestone is basic. So you have an alkaline solution there, right? We would talk about this being a high pH. And when we usually use the word high pH, that means the number is high. Doesn't necessarily mean because it's high pH, it means it's acidic. It actually means the opposite. It's basic. High pH means high number. And when we look at the high number, 9 to 11, that means it's basic. Okay. All right. So one other factor we look at as well is salt content. Salt content of an environment. And so salt plays a huge role in actually water as well. So salt and water, you add salt to an area, it's going to try to bind with water, or water will try to bind with salt. And we see this occurring in a lot of places that have a high salt environment. And because of that, we see organisms there. So a halophile is an organism that lives in a high salt environment, right? Can tolerate up to 35% salt content, right? That's a lot of salt, by the way. That is not the salt content of the ocean, in fact. The ocean is about 9 to 11% salt, something like that, which is a huge number. But we're talking about 35%. Excuse me. And for example, the Dead Sea in the Mediterranean and the Great Salt Lake have a tremendous amount of salt. And we see organisms living there as well. So we call these halophiles. Okay. What we call a facultative halophile is an organism that can tolerate a high salt environment but doesn't grow too well with it. So it doesn't prefer that area, doesn't prefer to grow in that area, but it will still grow, it just doesn't grow very well. We call this a facultative halophile. Staph aureus is actually a facultative halophile. So we find this, for example, in some humans on their skin, because the skin has a high salt content environment, top of the skin. So some of those organisms are found there. Staph aureus is found there, but most of us have Staph epidermidis. Staph epidermidis is a common, what we call ubiquitous microorganism that's found on skin. But some of our folks also have Staph aureus on our skin. Not all of us do, but that is a facultative halophile. Doesn't grow too well there, but it still can be found. Okay. All right. So this is what we talk about when we talk about salt and its effects on a cell, right? What we call plasmolysis, right, is the effect of a high solute concentration. Okay, so when we put a lot of, for example, salt in an area in water, we start to see the plasmolysis. We would talk about this being a type of tonicity, if you remember this word, right? And in fact, as water is going out of the cell, okay, so in fact, what you're seeing here, in fact, on the left side, there would be, in fact, two cell walls here. And because it is found, this will be in a high, let's say, salt content. I'll just put sodium outside. High salt content. It's going to cause water to rush out of the cell. because it causes water to rush out of the cell. What you see here, the effects are that the cell is losing water, and inside of itself, and this would be the plasma membrane, and the inside of the bacteria is actually shrinking. And so this little remnant here is actually the original cell wall. So it's shrinking, okay? And because of this, we see this in a higher salt environment where the cells will shrink. Again, they won't grow well, but they will still survive. Some of them will. Others do not tolerate salt whatsoever, and you add salt to the area, and it will cause them to not grow at all. Okay. Okay, so let's talk about oxygen, right? How oxygen is really important here. Many organisms live on the planet with oxygen, or with minimal oxygen, without oxygen, or minimal oxygen. So we see these places on Earth. We think mostly the Earth is, oxygenated, but there are places, for example, in the soil, that you don't have a lot of oxygen, and you'll still have growth there. There's places on our body, for example, and I'll show you a figure in just a second, where you don't have a lot of oxygen. Because you don't have a lot of oxygen, you still have microorganisms growing in these places, right? So for those organisms that don't live in oxygen, or with very minimum oxygen, right, oxygen can actually do interesting things and negative things. We always think of oxygen being a crucial thing that's important, but oxygen for some organisms is actually deadly. It's actually not good, right? When we look at oxygen, oxygen can actually diffuse across the plasma membrane pretty easily. Okay, so it doesn't need a channel on the cell membrane. It doesn't need a carrier to bring in oxygen. Oxygen will just diffuse or pass through the plasma membrane pretty easily. And so you can build up a lot of oxygen inside of yourself, right? This oxygen is actually converted into what we call a reactive oxygen species, a different form inside of a cell. Okay, so the oxygen, what it's telling you is that the oxygen turns into a different kind of chemical component, let's say. They can turn into what we call superoxide ions, okay, where they actually gain electrons, or they can actually turn into hydrogen peroxide. Yes, the exact same type of hydrogen peroxide, same chemical that you have, at your house, hydrogen peroxide that you use to clean wounds or dressings or whatever the case may be. Same hydrogen peroxide is what we're talking about. With the exception is this is a high concentration of hydrogen peroxide. The one that you have at your house, it's about 3%. Okay, we're talking about a much greater concentration of hydrogen peroxide. Both of those, superoxide ions and hydrogen peroxide molecule, can actually damage proteins and can damage DNA, both of which are bad for a cell. A cell will die if these are let loose and start to damage proteins and DNA, right? Because of which, organisms have actually adapted and have developed ways to actually detoxify, break these superoxide ions and hydrogen peroxide ions down, okay? So aerobic microbes, for example, ones that use oxygen for metabolism, that's what aerobic means. They need oxygen for metabolism, but they don't need oxygen to hang around in a cell, just kind of turning into these different components, but they do need it for metabolism to create energy. But what happens to the oxygen that's just hanging around? Well, that can be deadly, and these microbes actually use an enzyme to break down, or several enzymes, excuse me, to break down superoxide ion or hydrogen peroxide molecule. So we talk about these two enzymes. Superoxide dismutase is the enzyme that actually converts, for example, superoxide to hydrogen peroxide. Okay, so it breaks that down. It actually doesn't break it down, it adds it to hydrogen and turns it into hydrogen peroxide. Now, as you just saw, the list also includes hydrogen peroxide. So why in the world would you convert it to hydrogen peroxide if it's still going to do damage? Well, there's another enzyme. In fact, there's two enzymes. The next enzyme is called catalase. We'll get to know catalase pretty well because there is a type of test we can use for an organism to discover if it in fact does have this enzyme or it doesn't. And so we'll see that later in lab, so keep that in mind. But catalase is the enzyme that actually converts hydrogen peroxide, okay? And this is actually breaking down hydrogen peroxide into water and oxygen. And in that regard, it breaks those down, holds the water, holds the oxygen. If it needs water inside the cell, it'll keep the water. But if not, it will release it out of the cell along with oxygen. Okay, so again, oxygen turns into superoxide ion or hydrogen peroxide. We break them down, the organism breaks them down with these two enzymes, and then we release it back out. Okay, so what's an obligate anaerobe? Excuse me, an obligate aerobe. An obligate aerobe depends on O2 for cellular processes. And so you can write in parentheses metabolism, because these obligate aerobes need oxygen for metabolism. They actually need oxygen to make energy, right? Energy. Microaerophiles, and the key word here is micro. Micro means small. Aero means oxygen. Philes mean organism, right? Organism that uses a small amount of air or oxygen, right? Use only a small amount of oxygen. Okay, so since they only need a small amount of oxygen, they can live in a low oxygen setting, an area that has a very low amount of oxygen. That's where you find a microaerophile. Okay, and again, in reality, in the environment, you're going to find these places. And the facultative anaerobe, okay, is actually can grow, and facultative really means can grow in different places, right? So it can grow with and without oxygen. So when we hear the word facultative, that means with and without, so they can grow in both places, right? Facultative anaerobe, anaerobe means without oxygen, they can grow with and without oxygen, right? These two places, okay? And so they can actually use oxygen, but they can also use another process to create energy, and we call that fermentation. Okay, so they can switch using oxygen to fermentation, and both of which are totally acceptable. Okay, so this table, and I want you to make sure that you really study this table. Not that the table is going to be in the test. The table is used to study what we just talked about, oxygen use and the tolerance of oxygen. These help you to understand oxygen requirements or not requirements, or if they can tolerate oxygen or they can tolerate oxygen. So you see every single one of these, okay? Obligate aerobe, obligate anaerobe, microaerophile, aerotolerant anaerobe, facultative anaerobe, right? And because you see the growth in different parts of the tube, this is going to tell you if they use oxygen, they don't use oxygen for metabolism, or can they tolerate? Okay, so a good table to look at to review for these terms here. Okay, so here's your pathogens, right? Here's the different pathogens that you might find that are specific to different systems of the body, different places of the body, and you can see 5 here: lungs, blood and lymph, skin, stomach, large intestine. You can see some of the pathogens. Now, when you look at these pathogens and you look at where you find them in the body, you can also look at the oxygen level, right? And so that's how we kind of can start to look at these different organisms, these different pathogens, and look at where they grow because of look at the oxygen levels in those places. That's really what we're trying to talk about here. If you'll notice that most of those pathogens there, they're not obligate aerobes. Most of them are anaerobic, in fact, right? or they can tolerate oxygen. Okay, so it's very interesting to note that, as I mentioned, that most organisms are, don't need oxygen. They actually need a very small amount or none at all. And you can see this already evident in the places that you find these pathogens at, potentially. Okay, so microbes use the nutrients, right? So just to kind of recap, right? They use the nutrients in the environment. They use them to grow. They use them to build things to grow. And then eventually they use those to undergo replication. So again, that's the main topic here, what we're talking about, using these for their growth and then their cell division, right? Okay, so when we look at a cell, and if we take away water, about 90% of the cell's dry weight is these four elements, right? So we take away water and we dried out a cell. Most of it is carbon, hydrogen, oxygen, or nitrogen, right? And then the other 10% are made up of these different ions or different elements here, right? Different types here, and you can see the list there as well. Okay. So when we talk about essential nutrients, we're talking about the nutrients that are needed to build a new cell, right? The things that are absolutely needed to build cells, the parts of the cell structure, etc., right? And so some of those are organic and some of those are inorganic. As we mentioned in the list there right before, you can see that some of them are organic or inorganic, right? And again, what's organic, right? Anything that has these two elements in its chemical structure, right? Carbon and hydrogen. If it has one of each or more, it's considered organic. If it has one or the other, inorganic. If it has neither, inorganic, right? Okay. So we use the word macronutrients, right? Macronutrients are things that are needed in large amounts. For example, carbon. We need a bunch of that. And micronutrients are things that are needed in really small amounts. Okay? All right. So talking about nutrients, we can classify organisms depending on if they need, for example, a certain type of nutrient. And we classify that nutrient based on if it's organic or inorganic, right? So when we use the word heterotroph, they need some sort of an organic carbon, okay? Some sort of an organic carbon, right? Either it's going to be a carbohydrate, it'll be a lipid, or a protein, all three of those are organic carbons. They have organic carbon in them. They have carbon and hydrogen, and those are your macro nutrients or macromolecules, right? An autotroph, okay, does not need organic carbon, or at least doesn't need an external source of it. Doesn't need to get carbon from an outside source is what it's telling you. That's an autotroph, okay? What it actually does is an autotroph will use this process called carbon fixation, and it's a process that converts inorganic carbon into organic carbon, okay? So if, for example, this would be an inorganic molecule, right? What is that, right? Carbon dioxide, right? CO2, carbon dioxide. Notice it has one carbon molecule, but it doesn't have any hydrogen. Since it doesn't have any hydrogen, it's considered inorganic. Okay? And what would be organic? Let's just use that as our example, just to kind of keep it short. This would be methane, right? CH4, methane. Okay? Has carbon and hydrogen in it, be considered organic. So in autotrophs, they actually can use, for example, carbon dioxide, and can convert carbon dioxide so that way they can use the carbon for energy. So an autotroph does this, right? It can use inorganic molecules, carbon molecules, and can convert them, right? Convert them. Okay, so most of the nitrogen and the phosphorus can be taken from the organic nutrients. So when we see organic nutrients, when these organisms bring in organic nutrients that do like to use organic nutrients, they will usually pick nitrogen and phosphorus from the actual organic molecule. For example, proteins and lipids contain nitrogen or phosphorus, and for example, nucleic acids contain nitrogen. So they can pull those sources from those areas, right? When cells grab nitrogen directly from the atmosphere, we call that nitrogen fixers. Okay, nitrogen fixers. And we know for a fact that these organisms, these nitrogen fixators, these microorganisms that do this, actually extract them directly from the environment. Sometimes that environment is, for example, the soil. And I talk about nitrogen fixators because they're really important, because they'll actually take that nitrogen and convert it, and we use this, or plants use this, to grow from the soil. So they'll use this process too. Okay, and growth factors, right? Growth factors are things that are required from the environment that are needed for growth, right? So, for example, some microorganisms can actually build all the organic molecules that they need. For example, amino acids can't be built by a microorganism or a human. Or vitamins, some organisms cannot make vitamins, and so they'll need these from an outside source, from the environment itself, right? So the things that can't be made, that they need, that a cell needs to actually grow and reproduce, is what we call growth factors. Okay. So when we take those growth factors away, or if they're not present, then the organism doesn't grow. Okay. So when an organism needs many growth factors, we call that organism a fastidious organism. Okay, fastidious organism. All right, so when we talk about energy sources and we look at requirements of energy, one of them, as far as requirements of energy, are actually from different places, right? So if you're a phototroph, right, if you're an organism that uses light energy, then you are a phototroph. So some organisms, including bacteria, can actually use energy from light. So that actually means, energy from the sun. If you're a chemotroph, which most bacteria, for example, are chemotrophs, these are organisms that break down chemical compounds, molecules down, and when they break those down, they release energy. Most of those are going to be organic molecules or organic compounds. So when you use, for example, carbohydrates, and you break up carbohydrates, when you break those up, you release the energy. That is what we consider a chemotroph. Okay. So figure 7.11 is what you see here. This one is really important to actually study from, because this one talks about each type of energy source, right? So if you're a phototroph, you would use light energy, for example, sunlight. If you're a photoautotroph, that means you're going to use an inorganic molecule, and that's usually carbon dioxide and cyanobacteria, right? We call these blue-green bacteria, but they're usually found in fresh water. They use light energy, and their carbon source is carbon dioxide. And if you're a photoheterotroph, that means you use light from energy from the sun. You're organic, and for example, Heliobacillus mobilis is a bacteria that you find, for example, in water, that will use light energy, but then will also break down organic molecules to harness, to use for reproduction, right, for growth. If you're a chemotroph, and most organisms are chemotrophs, they will use nutrients, carbon sources, to break down, and will release the energy and use that energy for building things and for growth. And so when you are a chemoautotroph, your organic compound is usually carbon dioxide. It's inorganic. If you're a chemoheterotroph, this is the big, usually the big type of energy source umbrella where a lot of our organisms are found. You use an organic carbon source, and that organic carbon source, when you break open the bonds, will release the energy, and that's usually, for example, E. coli is found there, but a lot of your organisms fall in that group right there, chemoheterotroph. Okay. All right. So what we're going to do is really breeze through this one. We're not really going to go too much over this part of the PowerPoint, and we're going to kind of go forward past this point. And what we're looking at here is looking at the media, types of media, where it would be used, the names that we use for these types of media. And really what we want to do is we'll get into some of this, for example, in lab. So we'll see a lot of this in lab. I will talk a little bit about it at the very beginning, but I won't discuss too much past that point, and we'll kind of move on to the next part of the chapter here. Okay. And so I will talk about this one, figure 712, right? Growth media, right? There's different types of media, different types of components that we use to actually grow media, to cultivate to grow bacteria, to grow microorganisms, and to cultivate those microorganisms, right? And so we use different ones being liquid, for example, liquid media, solid media, semi-solid media. And you've actually, believe it or not, used all of these so far. When you inoculated bacterial tubes, for example, or the plates that you've already done, some of them were in a liquid, some of them were solid, some of them were the half and half kind of thing, where they were kind of a jello-like substance, not solid, not liquid 100%. And so you've used these, and we use these different things to actually look at different types of growth and different types of organisms growing, right? Okay, just really quick, just to kind of revisit this, we've seen this in the lab before also, right? So we know about this, agar, right? It's a polysaccharide, okay? And we use that, and we add liquid media to it, and that makes the media semi-solid. Gives us a nice surface for growth, for example, growth for example, in a petri dish, right? Again, we go back to the lab exercise where you took samples of your body, took samples of the environment, plates, right? Okay. So we can actually use solid types of media, and we can put it in test tubes like you see here, right? So this is a solid media. This is also a solid media also. And then depending on what kind of media it is, sometimes we can use a slant just like that. We can use a deep just like this. And we can test different things off of that, right? This test here, the Simmons citrate test, not that you need to know this for an exam yet, so, but a Simmons citrate test does have different types of nutrients and an indicator inside of itself, and it's colored green here, and will turn a certain color if it's positive. If the organism that we put the organism on this media, if that organism in fact uses some of the things inside of the media, an indicator, a color change will happen, indicating that it in fact uses some of those things from the media there, right? This one we call the motility test, actually indicates an organism being motile, movement. And how do we know it moves? We can start to see growth going sideways. So we used one of those inoculation needles, not the one with the loop on there in the lab. We would use a needle, and we'd stick the bacteria all the way down through the solid and watch to see if the growth was going side to side, right? And if it goes side to side, this would be a motility test. We would know that this organism is motile. It has flagella, for example, that make it move, right? So that's what we're testing for. It's really neat stuff. Okay, so we can classify media being complex and defined, right? A complex media, for example, on the left side, and again, this is just a recipe for a complex media, but a complex media basically has a lot of different things, but we don't know the exact levels of each one. Okay, it's just kind of we know that there's these nutrients that are really important, although we don't know exactly how much there is. That would be called a complex. And a defined media, so we know exactly how many things, what types of things are found for in this type of media. We call this a defined, or we know exactly what we have in that type of media, right? Precisely, right? Okay, so let me see if I can kind of breeze through some of this. We're going to kind of go past a lot of this here, because a lot of this, like I mentioned, we're going to go through the lab. So, or in it, we're going to go through it in the lab itself, right? Okay. Let's talk about this one really quick, this slide here. Talking about aseptic techniques. Want to revisit this, right? Again, methods that we've been using. Again, when I mention aseptic technique in the lab, it's really important because this is what we're seeing. So let me read this right here, just to kind of, hammer home the point, right? It's any method that we do that's designed to prevent not introducing contaminated microbes to a patient, to a sample, or to others in a healthcare setting. So again, we use aseptic techniques in a healthcare setting, but we also use them in the lab, and that's really what we're trying to practice, is these aseptic techniques, preventing cross-contamination, right? This is the key word, cross-contamination. So we protect people, promote the sample's integrity, making sure that that sample is the pure sample and not contaminated with other substances or other types of organisms, right? Okay, so again, when we collect samples, we use aseptic technique, right? Okay, so when we collect it, we have to transport it aseptically, prevent contamination not only of ourselves, of other people, of settings, of healthcare settings, places like that. We have to collect it using sterile materials, making sure that the sample doesn't get contaminated with the tools that we use to actually collect the sample from. And hand washing is super important. Hand washing, as we know, hands are a perfect place where we actually can contaminate other people, substances, objects, things of that nature. So hand washing is key there too, right? And again, just shows you some of the things. You wear gloves, you would seal the sample. The properties of where you took it must be held, must be taken in consideration, held for consideration. And then usually you're going to refrigerate it to stop growth or inhibit growth or prevent the organism from dying, expiring before you actually can test it, right? Okay, so we're going to talk about streak plating in lab. So I just want to show you what a streak plate is. This is figure 7.17 in your book. This is how you streak plate. The purpose of streak plating is actually to separate bacteria so you can see the colonies growing. The colony is one organism that grows and grows and grows and grows, forming a little circle or a little divot. And you see this in this part of the, excuse me, wrong color there. This part of the plate there, number 3 there, right? Number 3. Okay, we've separated it so much. We started at number 1, went to 2, and went to 3. We separated so much that we see individual colonies separate from the actual original sample, which is down here. This would be number 1. This is where we first introduced the microorganisms to our plate, and then we actually took some of it and went this way, and then took some of that and streaked it this way. And that's what you see there. That's a streak plate. And we'll get to know this. You'll actually do this technique and try to get good at doing streak plates. Okay. All right. So Just to kind of put in context, we can actually count how many organisms are growing on a culture. So we can track growth itself. We call this cell counting. We can do this through a microscope and actually count how many cells and do some math to actually look at how many of these organisms are growing in a given time. You can actually use this type of mechanism called a cytometer, and you can actually count how many organisms fall there, and then that's a way to count how many of those there. And we're going to try to do this experiment when we come back, hopefully the week after spring break is plate counts, actually counting how many plates and diluting the sample, and see if we can actually use this experiment. So we're seeing this here, right? 9-to-1 sample. So we're going to try to replicate this and see if we can actually use an indirect method of counting, right? Or sorry, the direct method of counting by diluting the sample. We call this dilution sample, right? All right, we'll continue to go on here. You can also measure it by shining light through it and having a machine read how much light goes through the test tube. That can tell you how much growth there's going to be or less growth, and this is what we see here. Okay, so let's just move on here to controlling microbial growth. Okay, so there's going to be a lot going on here in growth and in controlling growth, because there is a lot of what we call germicides, right? Germicides are any compound that we use to control microbial growth, right? Any compound, we call a germicide, is any compound that kills actually growing microorganisms. And most of those are going to be pathogenic, right? So that's our whole point. Germicides are trying to kill these organisms that cause then infections. We would, you know, in the short term, we would use the word germ, but germicidals are these chemicals that are killing pathogenic microorganisms. So we're trying to keep the growth at a minimum or not have growth at all, right? And we can see why that's really important, right? Okay. For example, they use this this example here, right? Water sanitation, right? To a hospital or cleaning in a restaurant are super important because we want to control the growth of those microorganisms. If those microorganisms, for example, are found in water and then we use that water in a hospital, many people will get sick. If we don't clean correctly, clean tables, clean floors, clean cooking equipment in a restaurant, clean the instruments, sometimes hands, that we use in a restaurant, we can also get people sick. So again, they are great examples of controlling microbial growth, right? So let's define these terms. Some of these terms can be really confusing to people, and some people have always thought that, you know, some of these words mean a certain thing, and come to find out they mean something completely different, right? So we'll first start with this first one, right? Decontamination, right? Decontamination is the process of removing or reducing populations of microbes to render an object, right? An object. That's the key word here. An object safe for handling. That's decontamination. Okay? Decontamination. Removing or reducing populations of microbes to render an object safe for handling. So you can decontaminate something, right? Decontaminate an object. Okay? By decontaminating an object, you've removed enough of the population of microbes that it's safe to handle or to use, right? Surgical equipment is a great example of that. You decontaminate, you remove the organisms, right? And then when you sanitize or sterilize, excuse me, use the word sterilize, you eliminate all organisms, viruses including, and endospores, right? In medical procedures that require sterilization, because you're introducing, sometimes those procedures introduce things to a sterile environment, the tools that you use or the equipment that you use has to be sterilized itself, sterile and sterile, right? And so you eliminate everything from that particular environment. So for example, when we sterilize test tubes, petri dishes, the media that we make, we're removing every single bacteria, virus, and endospore in a certain process that does that, right? Sterilization. Disinfection actually reduces the number of microbes in an area, right? Okay. So what's the difference between decontamination and disinfection? Pretty much the same, with the exception of decontamination means an object. It's really used for an object, reducing or removing populations. And disinfection is also used with objects, but things that we can consume or we actually put in ourselves, right? Cosmetics is one, disinfection. Foods, right? We can disinfect foods. Surfaces, that's an object. Medical equipment, right? Disinfection. Okay. So we're going to talk about physical and chemical methods of controlling microbial growth, and we'll start with physical here, right? Temperature changes, right? So we know that increasing and decreasing temperature, we've already discussed that in this PowerPoint and this chapter, that physical changes, increasing, decreasing temperature, can affect growth. So we know that that's one thing that we can use for controlling microbes. Radiation. Radiation plays a role in growth, right? Or inhibiting growth. And filtration. Filtering things out can actually be a method of controlling growth, right? Or controlling microbes, right? All right, so what do we talk about when we talk about temperature changes? Refrigeration, freezing, actually slow the growth of microbes, right? Slow the growth of microbes. So what that does is, if we're talking about food, it actually slows the spoilage, the spoilage of food, right? The spoilage of food. And that's what refrigeration does, right? Refrigeration does that. Freezing does the same thing, right? You can use it in a lab. You can increase the shelf life of media. You can slow the growth of bacteria in a refrigerator in a lab. Clinical samples, you can preserve your clinical samples and don't have them spoil as well, right? So most organisms are really sensitive to heat. Some of them actually thrive in heat, but most of them are sensitive to heat. And so we can use heat for sterilization or decontamination, right? So that's what we use there. So the term decimal reduction time or death time, we also know this is probably death time. It's the time in minutes that it takes to kill 90% of a population at a set temperature. So if I set the temperature at, I don't know, 90 degrees Celsius, which is really, really quite hot, 90 degrees Celsius, and I measure all the organisms at every minute, and let's say at 10 minutes, 90% of all the organisms already died, right? That would be our death time or our decimal reduction time in my example, right? So it would be 90 degrees Celsius at dermal death point and death time, right? Okay, so the temperature time and the death time, right? And so that's what we see here, right? Shortest amount of time at a certain temperature that needs to kill all the microbes in a sample, and then all the microbes at a minimum temperature to kill all the microbes within 10 minutes, right? So an autoclave is a machine that actually sterilizes equipment. We have an autoclave here on campus as well. That autoclave we use to sterilize all of our media. We also use to decontaminate and also to sterilize any equipment that's been contaminated potentially by organisms. So all your test tubes, all your plates, all have to get autoclaved before we throw them away or reuse the glassware for experiments. So we reuse all that stuff as well, and everything gets autoclaved. So an autoclave is important because it uses temperature, water, and pressure, right? And time, actually, if you look at the fourth one. Temperature, pressure, water, time. And we use all of those for autoclaving, right? So here's our standard for autoclaving. Make sure you understand this, right? 15 pounds per square inch. And so we don't adjust that in our autoclave. It's automatically will set its point for 15 pounds. And that's PSI, right? Pounds per square inch is PSI. That's our pressure. 121 degrees Celsius, 15 pounds. It's usually 15 to 20 minutes, right? 121, 15 pounds, 20 minutes, right? Is our standard time, right? For autoclaving. And most things will sterilize with that standard time. Now, if there's special things that we think are really important, like pyrons, for example, pyrons are notoriously hard to kill, right? To not kill, but denature. We would set that time for a lot longer. So it just depends, right? But most things are that time there. Okay. So we know boiling. Everybody knows that if you boil water, for example, if the water hasn't been treated, you would need to boil water to actually kill microbe growth, and then that would be safe to drink, normally speaking, right? And so we see this, right? Boiling is another way to apply temperature to cause microbial death, right? Or no growth. And so when we talk about pasteurization, pasteurization is actually a type of boiling, if you will, okay? And so we actually don't really boil the stuff, but we get it really close to boiling point. And it actually is a way we use it for certain amounts of food, like for example here in this figure, milk, right? Milk is used for pasteurization, right? And pasteurization was actually invented by Pasteur, right? Louis Pasteur. Louis Pasteur, the scientist with the swan neck flask, if you remember him from the earlier chapters in the book. Louis Pasteur actually invented the process of pasteurization, which is actually increasing the temperature of the solution, like milk, right before boiling and keeping it there for a certain amount of time to kill potential bad organisms and keep the good organisms alive, right? So it leaves the harmless microorganisms there, but kills the ones that cause pathogens. Now, you know where milk comes from. It would come from a cow, and it would come from a portion of the cow we call an udder, right? And the udder, in fact, is found in a really close place to the digestive system, where the cow basically poops and pees, right? A perfect place that harbors, for example, many of these, right? E. coli, right? Sometimes you have salmonella in that area, listeria, you know, those kinds of things. All of which get, because of pasteurization, get killed off. They die completely off of in the solution. But you would find those in milk. So if you take normal milk and don't pasteurize it, you potentially have those things because they're so close together, so close to the digestive system of the cow. And by pasteurization, that actually tends to inhibit those or kill those, in fact, organisms, and leave the harmless ones around. Now, the harmless ones are the ones that are normally supposed to be found in milk, the Lactobacillus acidophilus, those types of organisms that are found in milk that are beneficial or harmless, right? Dry heat does the same thing, except we don't use water. This is actually heat, right, with a waterless area, right? Ovens do that. Incineration does that. You can use that for sterilization or disinfection. Using your inoculation loop is a actual form of dry heat sterilization, right? So you burn it red hot. That's how you know that you've actually increased the temperature so much that you've killed whatever's on the surface and you've sterilized your loop. That's a great example of that. Radiation, high energy waves. Radiation usually comes from, for example, naturally from the sun, radiation. But we can also, we figured out actually how to synthesize radiation. We can use machines like X-rays, for example, give off radiation. And so we can use those as well. Machines like gamma rays or X-ray machines can actually shoot ionizing radiation. And what this actually does is kill the microbes, inactivates viruses, and what they do is they focus and damage the nucleic acids. Nucleic acids being the genetic material of a virus or a bacteria, right? We actually use this in foods, for example, and pharmaceutical industries. We use ionizing radiation. Right? Non-ionizing radiation would be like UV light, right? UV light. UV light, in fact, causes in the genetic material thymine dimers. So when you have, for example, a long strand of DNA, right? And let's say we've opened just one side of the DNA strand here, and on this end we have an A, let's say, for example, a G, a T, a T, let's say another A, just in our example. Well, when UV light shines through, for example, into bacteria, for example, anywhere where you see two T's put together like you do here, right here in the middle there, you're actually going to form a thymine dimer. And what happens there, and I'm not going to repeat all the letters, but in fact, both of those bonds form there and they stick together right in this part of the strand, right? So you see both of those thymine dimers forming one bond, and when the DNA tries to read and create another strand of DNA, for example, if it's growing, when it reads that, it stops the process, and when it stops the process, it doesn't make a whole copy of the DNA, and because it doesn't make a whole copy of the DNA, it does not grow, it doesn't replicate. So that's what UV radiation can do, right? Well, lo and behold, there are some equipment that you can use, like this light box here, that shoots UV light into the actual surface, and so you can actually disinfect, for example, surfaces like you see here. This is a safety cabinet with a UV light, and it's actually shining there to disinfect the surface. So if you're doing experiments here, this is a lovely machine to use, because you can do these experiments and then keep the surrounding area free of microorganisms, of contamination, right? Filtration. Filtration is actually using filters to send either air or liquid through to filter out any organisms, right? So there are some filters out there, both that filter liquid and air, that are so small that they can even filter out viruses. And so we use one, for example, in a hospital setting that we call a HEPA filter. You can also use these, by the way, in your house, because most of your air conditioning nowadays has HEPA filters attached to it, or you can buy HEPA filters for this. And that stands for high efficiency particulate air filter, and they have many small filters that actually filter out your air and can actually clean the air. And we use these in hospitals because if we've got a circulation system, an AC system, and we've got a sick patient in one room, that should be contained, because the filter, before you hit the air conditioning, that, let's say, feeds several rooms, has a filter that filters out, let's say, contamination from that patient, and separates that from the air conditioning to other rooms. So it's very helpful to have that. So HEPA filters get used a lot in hospital settings, right? And so you see a HEPA filter there. It kind of gives you the percentage there, 99.97%. That's, the fibers there filter out almost everything. Micrometer, that's a really small pore size, and it kind of, you can see it trapping then all of the, all the organisms in the air. So interesting stuff, right? Filters can also work for liquids, and so we see those as well. So we can use those as well, right? So we can filter out stuff there, right? These are called life straws. I don't know if you've ever heard this before, but this was actually created. This was invented by some really smart folks that have invented these filters that are really nice to have in these areas that don't have filtering systems for water. So there was a lot of funding from a lot of people. One of them being the from Microsoft, and I can't think of the gentleman there that I'm talking about, but the founder of Microsoft Bill Gates, there he is, and his wife Melinda actually put a lot of funding into research for these types of filters. They wanted to make sure that they're cheap, by the way, because yes, they can create a wonderful filter, but if it is super expensive and it can't be afforded by people, or it can be afforded to produce in large amounts, then what's the point? So these life straws were invented, and these were given out to folks, for example, in Africa that don't have filtering system for their water, and lo and behold, it saves a bunch of lives because they can actually filter out the water and filter out all the bacteria or the viruses and prevent themselves from getting infections. So that's what you see there is two two individuals there with their kids sucking up the water. So just like a straw, sucking up the water there and filtering out that water. And there's a filter attached to there, 0.2 millimeter filter, micrometer, excuse me, and it's removing the pathogens, which is really neat because you've got a lot of different pathogens that you find in water. For example, cholera is a huge one that you find in water, E. coli and what have you. So really neat stuff. Okay, so let's talk about these, right? Chemicals that that control growth, okay, are called germicides, right? Germicides that kill microbes are called microbiocidals, and germicides that inhibit growth are called microbiostatics. Microbiocidal means death. Static means to keep it at a certain level, right? Inhibit the growth, not completely kill the microbes, right? All those are considered germicides, right? What's a disinfectant? Okay, is a type of germicide used to treat inanimate objects, things that are not living, tables, chairs, cups, hands, even surfaces of hands, for example. A disinfectant, inanimate objects, right? Tables, chairs, etc., right? Antiseptics, right? Applied to living tissue. And I mentioned hands being disinfectants. So why are those disinfectants and not antiseptics? Well, you got to go back to your A&P, right? Your anatomy and physiology, and go back to the integumentary system, because the surface of our skin is considered, by technical standpoints, dead, right? It's dead skin. Since it's dead skin, we can use disinfectants to actually treat dead skin, right? So we can use disinfectants on our hands. But there's a caution there, right? You got to really be careful in using disinfectants on your hands, because right underneath, in the several layers underneath, shortly following, are living tissue. So some disinfectants can leach, can leak all the way to the living tissue and then cause bad things to happen, right? And so we would use antiseptics, for example, to living tissue. Living tissue, let's say, being inside the mouth, we would use an antiseptic, right? Okay. So germicides are actually ranked in three ways, you being a low level, an intermediate level, and a high level. Which one are you, right? Low level, intermediate level, or high level? A low level will destroy almost all bacteria, with the exception of, for example, mycobacterium like tuberculosis, some fungi and some viruses, but not endospores, right? So that's the low level. Intermediate level destroys all bacteria, okay? All fungus, all viruses, but not endospores. And high level destroys all microbes and endospores. So it depends on the level you're discussing here. Okay. All right. So here's a really important table. I want you to make sure that you look and you study at that table, because you're going to need to know all these germicides, right? All these germicides. And they're easy to kind of put together, because if you'll notice, there's the 7 families there. 7 families of germicides. And so by looking at the families, detergents, alcohols, phenolics, aldehydes, halogens, peroxygens, or peroxides, and ethylene oxide, those are your 7 families. Everything falls under those, right? Everything falls under those. And then you want to look at the mode of action, right? Detergents do what? Alcohols, how do they behave? Etc. Etc. And on, right? So that's what you need to make sure that you look at. So the table is really important to kind of study there, right? Okay, so I'm going to go past this point. They talk about each one of these, right? And I'll let you read through that yourself. So there are all of the ones that are on that table there and how they fall in. So if you just study the table, you pretty much can get most of this here. They just kind of expand on some of this stuff here, right? All right, so there are some factors to be considered when you use a germicide, because as you can tell, 7 families, there's a lot of germicides. So we got to figure out which one to use at what time and for what particular purpose, right? So again, what item are you using it on? How is it going to react with the to the germicide? What's the concentration of the germicide? How long the treatment it needs to last? What types of agents are you trying to control? If there's organic or inorganic matter. What happens if there's that chemical on an equipment? Does it react with the equipment? Is it a bad place to have it? And then is there toxicity from that germicide? So those are the things to consider when you're actually looking at which type of germicide to use, right? And so when we look at endospores are typically, and mycobacterium are typically hard to treat. They actually have a different method of action. So because they are structurally different, because they have different processes of forming these, we have to treat them differently. And these two tables show the different types of methods that work for those, right? Viruses, again, that's a different type of substance and agent, right? So we got to take different types of methods for that as well. Okay. So again, they talk about prions there and so on and so forth. And lo and behold, we're at the end of the PowerPoint here, the end of the chapter, and it kind of summarizes a little bit of everything that we talked about all in one here, kind of discussing all the way from microbial growth, generation time, all the way to the end, how we are using these physical and chemical methods to treat organisms. And so this is a nice kind of summary to everything. So if you get to a point where you're done with chapter 7 and you look at the summary at the end, it kind of just puts everything together. It's really neat to look at these kind of things at the end of the chapter, okay? Now at the end of here, he's got a clinical case. The clinical case is actually really nice to look at because it actually helps you to study what we're talking about in chapter 7. So I welcome you to look at that. So if you're at Saturday, Sunday, studying and want to summarize, want to tackle the case study, you can, and this is a really helpful thing for you. Okay, so I believe this is it. I think we're at the end of this chapter here, chapter 7. And again, I appreciate you guys listening. You know, I decided to do this recording. This is one of a couple of them I'm already doing for all of my classes. So I'm going to try to keep recording as much as possible. It does take a lot of time to do this, so I'm actually using a lot of time to actually record on top of actually doing lectures. So I'm trying to build on this kind of stuff on and on. So I appreciate you giving me some feedback. So I know this is kind of new for me. So this, you know, I did not practice this. I kind of just went off of it. And so I appreciate if you give me some feedback after the exam and let me know how you felt about this, if this helped you, if it didn't help you, what I can do to actually change this, if there's something that, you know, you saw in the video that you wanted to kind of, you know, if the highlighting wasn't working for you, if it did, or if you thought that this was good, or maybe you consider doing this. I'm very open to getting any kind of critique from you guys, because you're helping us, our department, you're helping our institution perform better and give you guys a better service. So I appreciate you letting me know too. It makes me a stronger professor. So please let me know what you feel about this video here, and if you like it, and hopefully I can continue to do these things for you. Okay, so let me know this weekend if you have any problems, if you have any issues, if you have any questions whatsoever with any of the chapters or anything like that. Please let me know, and I'll try to do my best to get back to you. Okay, thank you guys. I appreciate you listening in.